There we go. It's July 23rd, and it's our first in this year's series. We had a really good year last year with 13 lectures, plus one lecture about lab findings in the interim that Dee Burkhart helped put together. Thanks, Dee. I see you're on. I see a lot of familiar names and faces, so that's awesome that people are either coming back or friends from around the country are coming on to join us that we've interacted with at meetings. So it's really, really good to see folks coming on. A few housekeeping things. So overall, for anybody that hasn't been on the seminar series before, first, I'm Pete Baker. I'm the program director for MBG at University of Colorado. And this is the second year we're doing the series. Last year, again, we had 13 different lectures, very, very well received. And we've gotten a lot of encouragement to move on with this and to expand it. So this year, we have 21 lectures over the course of the next few months, so going from July into December. We also are trying to add a test at the end for MBG fellows and probably CBG fellows too, that should be should mimic in some ways the board exam to give you some practice for that. So we're asking from sample questions from all of the lecturers pertaining to what they're teaching, and we will have that at the end to your program directors and you'll be able to fill it out electronically on an honor system, of course, send it back to me. I can grade it and give your program directors a final grade for the lecture or for the test. Other things that we tried to do in the interim, we tried to get CME credit for all of these lectures, which would have been about 30 hours total CME credit. Our source for accreditation was the University of Colorado, and they fell through on us last minute. That's the long and the short of it. And we weren't very happy because it put us in an awkward position, not being able to apply for it on short notice from anywhere else. But currently we're getting a grant from Ultragenics to help pay for it, and also starting to apply for it through a couple different possible sources, so that by next year, this time, we should have CME established, at least for the MDs, if not for other folks who are on. So again, this is focused on MBG fellows and for MBG fellows, but we know from last year that we have a whole lot of other people on. And so we're very, very happy and welcoming to have GCs, RDs, APPs, other faculty, new faculty, and older faculty. I consider myself old, old faculty from lots of different places around the country, and actually from around the world. We have some international folks coming on too. So everybody's welcome. Whether or not you'll be able to get credit for this for CME depends on your specialty and whether or not we can obtain accreditation, but hopefully we'll have that for you by next year. Overall, the format for this, it's a lecture that's given this week by Dr. Falk from CHOP. I see Marnie, you're on. Thank you for joining us for about 75 minutes, and then question and answer at the end. Before last year, we had question and answer be routed through the chat and through me. I'd like it to be a little bit more organic this year and a little bit less formal. So at the end, we'll just open it up to questions. You can put them in through the chat and I can help moderate that, or you can speak up, then please feel free to do that. We're hoping to have good interactions both between the speaker faculty and the folks who are watching, but also between everybody who's watching. There's a lot of people on here with fantastic experience in the field, and so having that input is always welcome. I think, also, as last year, lectures will be available both through a Dropbox link sent out to the listserv, which is now, I think, over 350 people. So that'll be available to you if you're on the listserv. If you're not on the listserv, which I assume you're on it if you're on this call, but if you're not, let Holly know or I, and we can get you on. And if you know anybody who'd like to be on, send us their information too, or have them send us their information. But also the lecture will be available, the Zoom recorded lecture to watch asynchronously on the SIMD website. So all of that housekeeping is done. Without further ado, I'd like to welcome Dr. Falk, who will be talking to us today about mitochondrial disorders. This is one of several lectures on mitochondrial disorders over the course of the series. And if you want a full list of what we're going to be talking about for curriculum this year, you can go onto Holly's email that she sent out, and we have a full list of the curriculum for the rest of the semester. So Marnie, I will let you take it away. If you want to test out your screen sharing capabilities, I'm happy to help with that.
Okay, great. Well, it's a privilege and an honor to be here. I think I'm the first lecture, right, of the series. So this is
yes,
super exciting. I'm really, really impressed with everything the SIMD does, and Pete, of course, in arranging this series. And I just think the programs are terrific. So thank you again for letting me be your inaugural series speaker this year. Really neat. Can you see my screen?
I can, yep.
Oh, you know what? I hear like there's a background buzz. Let me just close the window. Hold on. Okay, is that better? Can you hear me now?
Yeah, you sound good.
Okay, terrific. Okay, so we'll go ahead and get started. And I guess, Pete, you want me to wait for the questions till the end? Is that your preference?
I don't have a preference. You can weave them throughout, or you can have them all at the end. It's up to you.
Maybe we'll wait for the end if that's okay, only because I'm not sure I can see them.
Yeah.
So I don't, but if there's something critical, please stop me, you know, and just talk. But I'm not sure I could read the chat while I'm on this full screen view.
Yeah, no, absolutely. That sounds good.
Okay, terrific. Well, I am again delighted that you're interested in, of course, and we'll be learning today about mitochondrial disorders. My disclosures are that I'm a scientific advisory board member on the United Mitochondrial Disease Foundation, the Genesis Foundation, and several companies. And I work as a research collaborator, consultant, or PI to develop therapeutics for mitochondrial disease. It's always good to start at the beginning. And so we'll go over first the definition of mitochondrial disease and the major clinical features. Most of you, hopefully all of you know that mitochondria are the powerhouse of the cell. I always say in my lectures, it is really the most significant educational achievement of our high school systems in America. There's very few, if any, people that graduate high school not knowing that mitochondria are the powerhouse of the cell, and then the knowledge falls off from there. So it is true that mitochondria are our cellular batteries. It's where we produce energy, and the more energy our organs need, the more mitochondria they have. From a more biologic perspective, they're cytoplasmic organelles. They are arisen from an ancient symbiont ancestor. So approximately 2.5 billion years ago, purple sulfur cyanobacteria invaded another cell and allowed multicellular life to evolve because now the cells could handle oxygen. And of course, the mitochondria make energy, but from a medical biochemical genetics perspective, it's really essential to understand mitochondria to realize how much else they're doing. They regulate calcium homeostasis. They're the site of programmed cell death initiation, or apoptosis. They're the major site in the cell where free radicals are both generated and scavenged. They are central to the first few steps in steroid biosynthesis. And very simply, if intermediary metabolism is an orchestra, mitochondria are the conductor. There's very few, if any, pathways that don't go through the mitochondria at some point. All that said, historically, mitochondrial disease occurs when this pathway fails. This is the electron transport chain, also known as the respiratory chain or the oxidative phosphorylation system. And it's the final common pathway to not making energy properly. It is the site where all of intermediary metabolism from a teleologic perspective is making reducing equivalents as NADH or FADH2, which gets shuttled either NADH through complex 1 or FADH2 to complex 2 within the inner mitochondrial membrane to coenzyme Q. Electrons are then shuttled to complex 3, to cytochrome C, to complex 4, and at that point, they breathe, or molecular oxygen is reduced to water. And this process of aerobic respiration couples the electron transport across the electron transport chain. So essentially 1 to 4 is considered the ETS, to a proton gradient that gets created by shuttling protons out of the matrix or inside of the mitochondria into the intermembrane space between the inner and outer mitochondrial membrane. That proton gradient specifically is made through pores in complexes 1, 3, and 4. And then it's dissipated when energy is needed to be created through the 5th complex, where the phosphorylation system known as ATP synthase, where adenosine diphosphate plus inorganic phosphate are coupled to make adenosine triphosphate. And that's energy, chemically speaking. It's high energy phosphate bonds that you break when you need them. And so this system, when it fails, and we'll spend the rest of the lecture talking about when and how and why it fails, that is a mitochondrial disease. The mitochondrial energy spectrum, I think, is helpful to think about for how mitochondria affect all of us. The mitochondrial disease patient is someone whose batteries at the start of the day are maybe at 20%. And if you ask a mitochondrial disease patient, they'll tell you that their life feels analogous to an iPhone, where when you look in that upper corner at your battery percentage, it really helps you understand what you can get done in the day. If you start out the day at 10%, you're pretty worried, right? Something didn't charge properly. And if you're at 5% at the end of the day, you wonder what you're going to be able to do before your phone shuts down. And that's a lot like a mitochondrial disease person's life. A lot of chronic diseases, though, have mitochondrial dysfunction. It's very clear, Alzheimer's disease and Parkinson's disease and diabetes and aging itself. And then all of us, have variable energy at different times in our life and at different times of the day, and the things we do, what we eat, and how we exercise affects that capacity. And even extreme athletes, they're always trying to improve their performance by increasing their energy. So what is a mitochondrial disease? Our understanding of what this is has changed relatively rapidly over the last decade. About 20 years ago, Doctors Munich and Rusten accurately described a mitochondrial disease as any symptom in any organ at any age by any mode of inheritance. And that was true and yet not helpful. So a lot of people started to think, is this real? Who has this? How can you tell when it's there? A big problem is that we do not have a common biomarker for mitochondrial disease. A lot of people want that to be lactate or pyruvate or the ratio. It's just not performing in the way that people want. A lot of people have tried, and there's some times where it is helpful, but there's many times where the lactate is elevated in non-mitochondrial disorders or where it's not elevated in definite mitochondrial diseases. Overall, there's about an 80% sensitivity and specificity of lactate as a biomarker for mitochondrial disease. What we now know also is that there's not just one genetic cause of mitochondrial disease. In 1988, Dr. Wallace, who's now at CHOP, discovered the first mitochondrial DNA disease. We now know of pathogenic mutations, at least 90 of them, across 37 mitochondrial DNA genes, and more than 300 nuclear genes have now been definitively associated with causing primary mitochondrial disease. Collectively, the minimum prevalence of mitochondrial disease is 1 in 4,300 people across all ages. And as we've learned more, we realize, as we've known, for the last 70, 80 years, is that mitochondrial disease is an organ disease. Organs fail, or more typically, multiple organs fail. And the basis why Peter Mitchell got the Nobel Prize was figuring out how the oxidative phosphorylation system makes energy, and that this is a problem. These are energy disorders. But more recently, because of the molecular genetics advances, we really understand this is really a series of pathways that become disrupted, each with unique molecular etiologies, and as we'll talk about at the end, different therapeutic opportunities. So what does a patient look like who comes into your clinic? Most patients with mitochondrial disease have a neurologic problem. But I like to think of us having multiple nervous systems: the central nervous system, the peripheral nervous system, the autonomic nervous system, even the lymphatic nervous system or the gastrointestinal nervous system. And so all of these organs can become affected. A lot of patients can have a range of problems that might be more specific, certain types of what we call metabolic strokes, or a stroke in a non-vascular pattern in a high energy demand region, such as sometimes the cortex, but more often the deep brain, so the brainstem, the midbrain, the basal ganglia, and a lot of the symptoms correlate. So balance problems, headaches, dementia and Parkinsonism can be seen, developmental problems in children, psychiatric or mood disorders, developmental regression. In addition to the central nervous system, peripheral neuropathy and autonomic dysfunction are quite common, and muscle problems are very common as well, often described as muscle weakness, lack of endurance, or exercise intolerance with fatigue. Patients often have sensorineural hearing loss. They may have any range of eye problems, eye muscle, eyelid, ptosis, or ophthalmoplegia, not moving your eyes. The optic nerve and the retina are often very commonly at risk or involved with dysfunction. And yet any other organ can become involved. Respiratory problems, cardiac problems, typically conduction defects and cardiomyopathies, liver problems and a subset, renal problems that can be glomerular or tubule, a whole suite of endocrine problems, which are often treatable, but really important to identify: diabetes, pancreatitis, adrenal insufficiency, and so on. A series of gastrointestinal problems, including failure to thrive, reproductive problems, and even things like scoliosis and short stature and bone marrow failure. The arrows, of course, are for us geneticists, who aren't always as comfortable looking at every type of imaging, but you'll see that there's often deep gray abnormalities on the MRI, evidence on eye exam of retinal pigmentary abnormalities. This is a classic hallmark when you look at a muscle biopsy, where when you stain, you see an increase of mitochondria along the borders of the muscle fibers. This is called a ragged red fiber. So essentially, most of the mitochondria aren't making energy, and so the body's adapting by trying to make more. They're still not functional, but you have more of them. That's a ragged red fiber. This is a ring sideroblast in certain types of anemias that you would see. This is an example on an X-ray of a patient with pseudo-obstruction, which can often precede a stroke in patients with MELAS. And this is reminding us of cardiomyopathies and muscle problems. What's remarkable is that on average, a mitochondrial disease patient has about 16 major symptoms. It's quite a lot. Most doctors are not trained to handle people with 16 major symptoms, maybe to address the top 2 or 3. And so a lot of patients have a hard time getting all of their concerns addressed. They also have a hard time sometimes being understood that this is a real problem, because they're often functional problems, not structural problems. Although, of course, some of these objective findings can occur. And I explain this to the patients by saying, if a child brought a doll to you and the doll's arm had fallen off, you knew there was some sort of trauma. But if the child brought the doll to you and it wasn't walking, it wasn't talking, the lights weren't flashing, most of us would say, let's try changing the batteries and see what happens. Because if the batteries don't work, nothing's going to work. That's really more like a mitochondrial disease. Think of it like the whole body's at risk because the energy supply is low. This is worth mentioning as a specific call out of a pediatric manifestation of mitochondrial disease. Lee syndrome was first identified in the 1950s by Dennis Lee, and it was an autopsy diagnosis of maybe a child who was okay in the first year of life, and then around a year and a half might have regression and quickly progress to demise. And an autopsy would find hyperintensities again in the deep brain, so in the basal ganglia, the brainstem, the midbrain that were bilaterally symmetric. And if you were to look on MRI, which now is the way we diagnose this rather than ideally on autopsy, we do see hyperintensities. They may fluctuate over time. And if we do an MR spectroscopy, where we put a voxel over the brain region with the signal abnormality, we often see abnormalities like this, which is what we call the bunny ears, or the peaks. They may be at different spots or up or down, depending on the x-axis TE time, but essentially this would be lactic acid would be building up. You have to be careful because some MRSs are just very sensitive and can detect lactate, and some people have high lactate if they're having seizures, and it may not be mitochondrial disease. But putting the whole story together, this is very concerning when you have a neurodevelopmental regression and metabolic strokes. Remarkably, we're now over 95 genetic causes. We just finished an international curation effort, and we're up to 113 genes in which mutations cause this problem. And you can go to the website of a resource called mseqdr.org that we coordinate, where you can see all the different genes and variants that might cause Lee syndrome. And you can link to the ClinGen website where more information is provided. We did a survey several years back. Dr. Zolkoply Cunningham was the lead author, and we were able to ask patients of all of their problems, like what is the most common, regardless of the specific gene, because it's so complicated. How do you make it simpler to think about these patients and their desire of where they want to be treated? And so it turns out, again, patients had 16 major problems. Some had just one, some had 35, which was the top number we asked. And yet of all of them, more than 95% had muscle weakness, chronic fatigue, and exercise intolerance. Gastrointestinal problems and balance problems were also very common. And you can see that there was a whole range of other problems. So seizures happen, but in about a third. Stroke happens, but overall in less than 20%. Same with liver and kidney disease. And it's really interesting because when you ask the patients, they tell you they really want treated, not the things that we might think they want treated, like diabetes or kidney disease or stroke, but the things that impact their life, which is weakness, fatigue, and exercise intolerance. A lot of people have thought about developing simplified clinical diagnostic criteria for mitochondrial disease. And you can see these were in 1996 through 2006. The problem with some of these is that they're very long. One of them is about 40 pages long, and they're very complicated to complete in a clinical setting, when there's limited time. The other problem with them is that they only can diagnose the types of mito disease that were known at the time they were created. So more than half of the genes that cause mito disease have been identified in the last 10 years. So a lot of them may not even have been identified by these criteria. Further, the categorizations were unsatisfying for patients. They were told they had definite disease, probable disease, possible disease, or unlikely. And this was really stressful. How do you walk around telling people you have possible disease? It's like saying possible pregnancy or possible diabetes. It's not really a diagnosis. So the consensus from the Mitochondrial Medicine Society has been to really change this now to be more about definite, suspected, or unlikely, to allow people to get past the diagnostic uncertainty. As I mentioned, there are no common biomarkers in the blood or the urine. We mentioned lactate. FGF21 and GDF15 have use, but it turns out it's not in all types of mito disease. For example, the GDF15 seems particularly high in a type of mitochondrial DNA disease known as MELAS. The FGF21 seems particularly high when there's mitochondrial depletion disorders, and so on. And there's also other reasons why these markers might be high, such as just having kidney disease or heart disease or pregnancy, and so on. Exercise testing has a lot of potential. Not every site has it, but it's really great because you can exercise somebody on a recumbent bicycle or a treadmill and monitor their gas exchange. And then based upon that, you can calculate non-invasively how well they are utilizing oxygen. And so you can actually measure their anaerobic threshold and something called their VO2 max, and these are low in mitochondrial disease. There's a host of other analytes that Dr. Haas at UCSD was the lead author more than a decade ago, and I highly recommend this paper to really describe when and how different amino acids or organic acids or acylcarnitines might be elevated. So for example, alanine is often high as an indicator, sort of like a hemoglobin A1C is to a glucose, that if your pyruvate and lactate are high over time, your alanine becomes high. It's also become clear that your alanine ratios, meaning alanine to lysine, should be less than 3, alanine to phenylalanine plus tyrosine should be less than 4. And these often are high in people with definite mito disease as a sort of soft sign. You'd also see high anion gaps in people that often have high lactates or high alanine. And that's sort of a poor man's way to guess the lactate's going to be abnormal. And then there's a range of other amino acids that may be high, but often aren't. In the urine, you can see a lot of different breakdown products, Krebs cycle intermediates, ethyl malonic acid, 3-methylgluconic, or dicarboxylic acids, but their absence doesn't mean that you don't have mito disease. And we've now realized over time that there's a lot of different ways to get these in your urine. And so it may not point you to a specific etiology. There's often secondary fatty acid oxidation disruption. And so looking at acylcarnitines can be helpful. Historically, from about the 1950s or 1960s, the test to diagnose mitochondrial disease was a muscle biopsy to be able to look at either polarographic or enzymatic activity of the mitochondrial function. So polarography currently isn't widely available around the world. And the center that was doing it, Center for Inherited Disorders of Energy Metabolism in Muscle, is no longer doing it. Our site and others will be setting up cellular assays where this can be done shortly. But historically, this is looking at the integrated function of the respiratory chain to breathe, because more than 90% of the oxygen that we inhale is actually utilized by the respiratory chain as an electron acceptor in complex 4. So it's definitely useful if it's low, but it's also can be very variable in the exact level within a pretty wide range. What's more commonly used, because oxphos needs living, fresh tissue, so it can't be done on frozen specimens, is enzymatic spectrophotometric assays, where you look at just complex 1 activity or just complex 4 activity, or maybe complex 1 to complex 3. There's been debates in the literature for a long time about whether it's more reliable in fresh versus frozen tissue, but the story ends that most people utilize frozen tissue because it has to be shipped somewhere for analysis. It's also important to recognize that oxphos may be normal and enzymatic activity is abnormal, and vice versa. So, it's really a descriptive finding. And historically, if an ETC enzyme activity is less than 25% of a concurrent control mean, then we think that there's a problem that's truly primary within the mitochondria. So let's jump into diagnostics and etiologies. The lingo can be confusing, so I thought we'd just address that first. In medical biochemical genetics, we're primarily dealing, at least at the current time, with primary mitochondrial disease. But in the greater world of medicine, secondary mitochondrial disease is much more common. So primary mitochondrial disease is where there's a genetic-based cause for the mitochondria to fail. It could be in the nuclear genes, it could be in the mitochondrial DNA genes, but the cause is somewhere in the inherent functioning of the mitochondria. And interestingly, more than 95% of all the genes that have been shown to cause a mitochondrial disease encode something that functions in the mitochondria. So not everything, but most. It's also important to realize these diseases are often chronic diseases, or maybe no symptoms until a stressor is experienced, fasting or an infection, or some other major stress. A secondary mitochondrial disease means there's nothing wrong inherently with the mitochondria, but there's something else going on that disrupts their function. And so acute or chronic, this could be acute or chronic, like a toxin or a drug. As a matter of fact, NCATS has a whole library, the National Center, I think, for the Advancement of Translational Science, where they study every drug to understand how much mitochondrial dysfunction it causes, because so many of them do disrupt the mitochondria. It could be environmental exposures, like when you garden, there's chemicals like rotenone, which are toxic poisons to the mitochondria. Aging itself impairs mitochondria by building up mitochondrial DNA mutations over time, and so on. It doesn't mean to say that it's not real. Once the problem is there, the mitochondria are not functioning and the people can have symptoms from it. The question is, what can we do about it? And everybody recognizes that when we have therapies for primary mitochondrial disease, they'll probably have a big impact in individuals with secondary mitochondrial dysfunction as well. When you think about primary mitochondrial disease, which is where I'll focus the rest of our talk, the likelihood of finding a mutation in either genome, you'll remember that most genes are in the nucleus, all 3 billion letters in a haploid genome, whereas the only organelle that has DNA outside the nucleus is the mitochondrial DNA, and it only has 37 genes. It's only 16,569 base pairs. So it's tiny. We tell our patients it's like a pamphlet where you have multiple copies, whereas the nuclear DNA is really sort of like an encyclopedia set, one per cell, and the mitochondria have lots per cell. But when you want to think about where's the etiology, it used to be thought that if you were a baby with a mitochondrial disease or suspected mitochondrial disease, that 90% of the time it's going to be a nuclear gene. And if you're an older person with problems, 90% of the time it's going to be a mitochondrial DNA change. As testing has gotten better, it's still this relative trend, but we now know it's more like 2/3, 1/3. About 2/3 of babies, it's going to be nuclear, and 1/3 is going to be mitochondrial DNA, and vice versa for older people. We're going to spend some, probably the most time, because it's unique to this lecture, talking about mitochondrial DNA disorders. So I mentioned the mitochondrial genome. It's is inside the mitochondria. It's double-stranded. It's 16 kb. It makes 37 genes. And what's fascinating, to me at least, is that it makes 13 polypeptides. Those are the 13 mRNA genes. And every one of them is a subunit that's very lipophilic at the core of the electron transport chain. 7 of them are in complex 1, 3 are in complex, 1 is in complex 3, 3 is in complex 4, and 2 are in complex 5. And what you'll notice is that complex 2 has none of them. And for this reason, the biochemists really argue if complex 2 really is part of the electron transport chain, because complex 2's other name is succinate dehydrogenase. So it's really a part of the Krebs cycle that feeds electrons in to the electron transport chain. The mitochondrial DNA also has 22 transfer RNAs and 2 ribosomal RNAs, and their whole job is to help translate these 13 polypeptides to be core subunits of the electron transport chain. And that's why if you have a mitochondrial DNA deletion of lots of genes or a point mutation, you're going to disrupt the electron transport chain or respiratory chain function because you're not making something that's essential for its core function. If you go to MitoMap, again, that Dr. Wallace has been running for 3 decades now, with Marie Lott, a scientist who has really manually curated every single variant. There's morbidity maps where you can see, along the 2 strands, there's a light strand and a heavy strand, as well as a D loop. The D loop has a lot of the kind of variation between populations, but not the disease variants. And you can see outside of it, within the different genes, where the different genes are and where some of the common mutations are. The mitochondrial genome has unique features. There are no introns. We tell patients all of the real estate matters. It does not undergo homologous recombination. It is not involved in meiosis. Its replication is continuous. It is not synchronized with the cell cycle. And relative to the nuclear DNA, the mitochondrial DNA has a high mutation rate. I believe at least a log or 10-fold higher rate. Mitochondrial DNA does interact with some proteins like TFAM in a nucleoid, but it does not have histones to protect it. And it's also important that there's 3 types of errors in the mitochondrial DNA that can occur in different tissues and have to be thought of individually so that you're sending the right tests to figure out if people have these types of problems. So the first is a point mutation. Here you'll have a single base pair or a few nucleotides changed. The second is deletions or duplications of large chunks of the mitochondrial DNA. So there's a very common 5 kilobase deletion, but we see deletions, a couple kilobases, 1 kilobase, 8 kilobase, and there's a whole program, a software program out there called MitoBreak, where you can go and look at all of the deletions that have been reported. Duplications are much more rare and much more difficult to detect. And then the third type of mitochondrial DNA problem isn't anything inherent in the genome. It's not a point mutation. It's not a deletion. It's actually depletion or proliferation. You actually have either too little or too much. And normally when you see that for depletion, it means there's a nuclear gene problem. There's something wrong regulating the amount of mitochondrial DNA. When you have too much mitochondrial DNA, it's often not a disease. It's an adaptation that the body is trying to respond to a problem. A key concept that will definitely be on your boards, no matter when or where you take them, is that mitochondrial DNA is maternally inherited through the oocyte. So that males and females can both be affected, but males do not pass it on because the sperm has mitochondrial DNA, but it's actively excluded from the resulting embryo. And there have been a few rare case reports dating back to 2003 in the New England Journal of Medicine, where there have been examples where some variants have been found to be inherited from the father, but most of these have not been replicated. And for the most part, we consider mitochondrial DNA to be passed on through the oocyte. And you can see here, this is an extreme example where every female passes it on, but more often there's about a 50% chance if the mother is symptomatic of the offspring also showing, inheriting the mutation to show symptoms, and about a 4% chance if the mother is asymptomatic, but a sibling had had it before. When you look at the mitochondrial DNA genome, you have to keep in mind that there's a lot of information in it. And when you compare a genome in a patient to a reference, you have to think who the reference was. And that's because mitochondrial DNA is actually used to track human migrations across evolutionary history. So until about 50,000 years ago, all humans were in Africa, and there's actually more variation among the original L haplogroups in Africa than among all the lineages that have left since. But what you can see, and again, Dr. Wallace traced this over time, Dr. Schur also made major contributions and others, is that there are fixed homoplasmic variants in the genome that are based upon evolutionary maternal lineages, such that the number of fixed homoplasmic mitochondrial DNA differences between any two people indicates the time since they shared a common mother. So if you're looking at somebody in your clinic from Asia and they have differences from the reference, they should have differences from the reference because the reference happens to be a Caucasian male from Europe. And so they're just having a variant does not mean it's pathogenic. So especially when it's fixed, and it's what we call haplogroup defining, the set of fixed variants that we think helps define your energy potential or your ability to convert nutrients to ATP as opposed to release it as heat through uncoupling of the electron transport chain. Those are not disease causing. That said, there's a lot of literature that certain other variants in the mitochondrial DNA might only manifest in a certain haplogroup background. So it is very important to understand sort of the origins of your patient and also what is a haplogroup defining variant versus not. Another essential concept to understand mitochondrial diseases caused by a problem in the mitochondrial DNA is heteroplasmy and threshold. These are unique concepts to mitochondrial DNA that are not relevant to nuclear genes. And they stem from the biologic principle that there are lots of copies of mitochondria in each cell, 100 to 1,000. As a matter of fact, the oocyte has something like 250,000 copies. It's a sack of mitochondria. Whereas per mitochondrion, you don't just have one mitogenome, you have multiple. And so it's really a stoichiometric math problem here, is that if a mutation becomes present in a certain subset of the mitochondrial genome's population in a given sample, a given cell, a given tissue, a given organ, it's not in all, it's just in a percentage. And that would be called heteroplasmic mutant. So heteroplasmy means there's two different populations. One of them is wild type or the reference, and the other is mutant. Homoplasmy means you only have one genome present in that population. It's either mutant or it's wild type. So you can be homoplasmic wild type, homoplasmic mutant, or heteroplasmic. And most disease-causing pathogenic mutations are heteroplasmic. So you have a mix where the higher the level of the mutation, or the corollary, the less of the wild type that you have to make these essential subunits of the electron transport chain, the more likely you're going to show clinical problems. And that's related to the threshold effect. So this threshold effect is specific to heteroplasmy, and it's when you think about each given organ or tissue and what is the level of the mutation that it can handle, or the lowest amount of normal mitochondrial DNA that it can tolerate before it shows symptoms. And this depends a lot on the organ. It depends a lot on the mutation. How badly is it affecting the protein? And of course, the level can be variable in different tissues, as we'll further explore in a minute. And so these two principles make mitochondrial DNA genetics fascinating, but complicated. How did this happen? Why is the stoichiometry, so reliably complex? So when we think about the maternal oocyte, it turns out that most mitochondrial DNA mutations arose in the oocyte of the individual you're caring for, while they were an egg inside of their mother, while her oocyte was being, while her gonads were being formed. So these are very early events in most cases. And it might just be that a population of that oocyte or the progenitors carried the variant. And let's say the mutation or the genome that has the variant, whether it's a deletion or a point mutation, is in red, and the wild type is in green. And of course, the nucleus in the cell is shown in blue. What happens when you actually get to now the oocyte that made your patient is that there was a mix of normal and abnormal. But whatever that mix was, it's hard to predict, because you go from about 250,000 mitochondria down to just a few hundred as that oocyte gets fertilized and divides. So it goes from 1 cell to 2 cells to 4 cells, and they're not making more mitochondria. They're just dividing the mitochondria that oocyte started with, until about day 3 or so. At that point, you go through this bottleneck. So the minimum number of mitochondria. And at that point, there's a progenitor cell for the embryo versus the placenta, and you have to just guess what the percentage was. So maybe the percentage here was 10%, but it became enriched, after the threshold, or maybe it became diluted. And the same thing happens as you go to the organ progenitors, so that you might wind up in a given individual with some organs that have a very high mutation load, let's say the brain, and some other ones, let's say the blood, where it's very low. So it's very challenging to look at the results from one tissue and understand what it was way back when, in the earliest stages, or in the oocyte. And so this is the demonstration of heteroplasmy, so having a mix in a given cell, and then the threshold, where the more of the mutation and the less of the normal, the more likely you are to have a clinical problem. It also reminds us why we need to test more than one tissue, because we really biologically need to assess what happened and where the problem might be. I always like to give case examples. So here's one. This was a child I was called to evaluate at the age of 19 because of cataracts. And you wouldn't think cataracts would be the primary presenting problem for a mitochondrial problem, a mitochondrial disorder. But when I took a history, I realized it wasn't. He had been seen by every specialist at our institution for cardiac problems, neurologic problems, endocrine problems, ophthalmologic problems, muscle problems, and lactic acidosis with high alanine. The reason why they were calling a genetics consult finally was because he had a pigmentary retinopathy that took out his peripheral vision. And now with the cataract, he was losing his central vision, and they wanted to know, what could do this. That now there was an urgency. He wound up getting a muscle biopsy and had these ragged red fibers. He also had a classic hallmark feature called Cox negative fibers. This is a double stain with the Cox and the SDH. So Cox is a complex 4 subunit. That's mitochondrial DNA encoded. SDH is a nuclear subunit that's nuclear encoded, of complex 2, forgive me. And what you can see is that it's here you see all blue, but here you see blue peeking through the brown. And I think that's telling us that there's something wrong with the mitochondrial DNA subunit, because it should all be brown unless the Cox, if there's less. We went on to actually sequence his entire genome. This was pretty revolutionary. And Li-Jun Wang, who we recently lost, unfortunately, who's been at Baylor and was just, revolutionary in this field, helped us actually sequence the child's cataract. And the cataract had 100% of this novel variant at position 12,264. And at the time, people weren't always doing whole genome sequencing of the mitochondrial DNA. They were just looking for the common mutations, things like MELAS and MERF and NARC that I'll come back to. But what was interesting is that it was 100% in his cataract. We did it on the muscle biopsy. It was 100% mutant. And when we looked in his blood, it was only 34% mutant. And this to us, in his cheek swab, it was also 100%. It was very helpful, because when you see heteroplasmy in some tissues, but homoplasmy in the affected tissues or higher heteroplasmy, that is consistent with a variant that's more likely to be disease causing. His father did not have the mutation, and his mother was deceased, but his grandmother, when we checked her, Dr. Wang actually helped us figure out that the mother's mother had the variant at 1% in her blood and at 18% in her cheek swab. So the grandmother was perfectly healthy, but had clearly transmitted this variant to her daughter, who had also appeared to be healthy, to the grandson, who you can see had it in every organ to different levels. So what about prevention of mitochondrial disease, pre-implantation genetic diagnosis? Once you know a family has a pathogenic mitochondrial DNA mutation, can you test for it the way you would for a nuclear gene to identify, which embryos might harbor it, might not, and select which ones to pick? For a long time, the answer here was no. Because the testing wasn't reliable from a sequencing perspective on one cell or one blastomere from an embryo on day 3 after the bottleneck occurred. However, over time, countries in Europe primarily have been pushing forward this effort in England and in the Netherlands, and they've and in the research world, and they found out that less than 5% heteroplasmy in the blastomere of the embryo should be unlikely, or should be very low likely, for the eventual child to come from that embryo to develop a clinical disease. It's still, of course, a little nerve-wracking because, as you saw, things can become enriched over generations, or even within a given individual, depending on the mutation. So a lot of people are sort of uncertain what level should be selected. Currently, the European groups are going up to 20%. Sometimes even a little bit higher, depending on the mutation, to select embryos to put back in. In terms of the child who's born after PGD, it's important to realize that some mothers who carry these mutations and are symptomatic have reduced fertility, so they might have to go through a lot of cycles and not even find an embryo that meets the criteria. But whether the child has a disease will depend on what the mutation is. And it also depends on the reliability of the method that was used to make that determination. Ultimately, we recommend that any child who is born following PGD is followed long-term in a mitochondrial medicine center, and also recognize that some variants don't present symptoms until adulthood. And so things like diabetes, hearing loss, headaches can be seen, and families need to be counseled about that. There's a methodology that got a lot of press a few years back, maybe 5 years ago now, because it was approved in England, in the United Kingdom, for clinical use. And it's being used there with individual licenses. When you know that a baby has died from a mitochondrial DNA disease, the parents have an option to request a license to have their embryos in the future undergo this pronuclear transfer procedure. What happens is that the intended mother has her nucleus extracted and saved, and an unrelated donor gives an oocyte where the nucleus is discarded, but the mitochondria that should be healthy, because it was the intended mother's mitochondria that had that mitoDNA mutation, the donor should have healthy mitochondria. So they actually fuse the donor's mitochondria with the intended parent's pronucleus within like the first 12 or 24 hours after fertilization. And then they reconstruct a zygote, and then that's what they implant. And so there hasn't been a lot of reporting on how this works. In America, this isn't legal. You can make the embryo, but there's a congressional rule, law, that you are not allowed to implant it. So it's still sort of on hold. But we have figured out ways, and I'm happy to talk if you have cases where we can send embryos for PGD, where the embryo stays in the United States, but we send the blastomere to the United Kingdom for analysis without the family having to travel there, which is very stressful for families to have to go internationally to get these procedures done. So nuclear gene disorders, I'll jump into now that we finished the mitochondrial DNA topic. Nuclear gene disorders are more run-of-the-mill, but there's a lot more of them as a cause of mitochondrial disease. So it's actually more mitochondrial diseases that occur from nuclear genes than from mitochondrial DNA genes. Why? Because mitochondrial DNA has more than mitochondria has more than 1,500 protein products. The mitochondrial DNA only makes 13. So obviously, right, the rest of the candidate causes are somewhere in the nuclear genes. And this says 250, it's over 300 now, nuclear genes implicated in mito disease. And we discover about 20 per year as an international community, at least 20 per year. So there's just an enormous growth rate in our understanding of these disorders. So when you think of a nuclear gene cause of mito disease, any inheritance pattern can be seen. Let me repeat that. Any inheritance pattern, autosomal recessive, autosomal dominant, X-linked. If it was a maternal inheritance, of course, that would be a mitochondrial DNA cause, but it can be confusing from small pedigrees to guess. What types of genes cause these different nuclear genes? They often encode proteins that are subunits of the OXPHOS system. There's about 81 subunits. And so all of them are fair game. Most of them aren't seen because they would be thought to be embryonic lethal, but that's where a lot of the causes are in the 5 complexes. OXPHOS biogenesis, so actually making the OXPHOS system or regulating it. Mitochondrial DNA, it turns out, is sort of like international business. It really needs a lot of things imported to function. The mitochondria doesn't make its own DNA. It actually needs to bring in nucleic acid and convert it to the right form and then transcribe it and translate it. And anything wrong in that process can be a mitochondrial disease. And the membrane dynamics is also a common cause of disease. So mitochondria have to interact with one another. They fuse their membranes, they break apart, they have to donate and repair themselves. And there's a lot of biochemical pathways too that can become disrupted. So is there one cause of mitochondrial disease that you should think of? People about 15 years ago thought it should be pol gamma, because pol gamma seemed like the most common cause that was known. We started having clinical testing for it around 2006. And all of that showed that approximately 3% of all mito diseases are caused by pol gamma variants or pathogenic variants. Still rare. Most mitochondrial diseases, are not common. And so the most common is pol G, but still rare. And this is just provided. You'll have access to these slides to remind us it's really important to be able to do counseling to know what's the inheritance pattern, what's the genetic cause, and then you can also predict recurrence risk to the sisters and brothers of the proband, and then also the offspring, whether the affected individual is female or male. Remember, only mitochondrial DNA diseases can't be passed on through the males, right? The other ones certainly can. We published a review that might be helpful to help think about a more simplified diagnostic algorithm. Everything always starts with a high index of suspicion. So when you're in clinic and you are thinking about the problems, whether there's a red flag symptom or just multiple non-specific symptoms, it all comes down to doing a really good clinical assessment and history, review of systems, and exam. And then you want to do a very careful pedigree. What you're looking for is often vaguer or maybe not exactly the same, but spectrum symptoms and relatives. Maybe somebody had a child with Lee syndrome. Did the mother have migraines? Did somebody have chronic fatigue? Did somebody have multiple sclerosis or non-specific other symptoms that might make you think there was an energy problem that maybe was not diagnosed? And then you want to phenotype whatever symptoms they have. If they have hearing loss, heart problems, kidney problems, you want to better understand what type. And you also want to just generally screen most patients when it's suspected for hearing, heart, and vision, since those are 3 organs that are very commonly abnormal in mito disease or can develop symptoms over time. We still do the biochemical screening in blood and urine I mentioned, and we also screen for problems that we might treat. So maybe they're anemic, maybe that's why they're tired, or maybe they have vitamin D deficiency, or maybe they have adrenal or thyroid insufficiencies. All of that can be treated. And they could be secondary to mito disease, but nonetheless problematic. And then we now very early on, before tissue biopsies, we utilize genetic testing. Most labs now that do exome sequencing will also offer concurrent mitochondrial DNA sequencing if you remember to request it and check the box. And in many places, there's no extra charge. Whole exome can be done on any tissue, blood, buccal, whatever the lab accepts. Mitogenome, you might need to be more thoughtful about where the symptoms are. And so, for example, in an older person, the MELAS, mitochondrial encephalopathy, lactic acid and stroke symptoms or syndrome comes from most commonly in 80% of cases a 3243 A to G variant, but the level disappears over time in blood. So in that situation, in an older person, you might want to check urine. And we've had cases where we can't find it in urine, and then we find it in muscle. So we only go on to tissue biopsy if we can't find it in the peripheral tissues. The other thing is sometimes you might find a really low variant, 2%, 4%, and you're like, is this enough of a heteroplasmy level to really cause disease? And then you might actually have to test more than one sample because of the issues of, biologically, the variant may not be in specific tissues just because of the origins of heteroplasmy. Or you really want to look to see if it's more common in one at a level that would make you think it could be disease causing. But there are some mutations we know at 20, 30% in blood reliably are the cause of disease, where there's others where it wouldn't be. So it might be mutation specific. And then once we know they have a mutation, then we have to provide care and offer them clinical trials. Whereas if it's negative, then we might go on to more tissue-based testing of the amount of mitochondrial DNA, the sequence of the mitochondrial DNA. At our institution, we developed a test where you look at the sequence, and if you find a deletion, you do digital droplet PCR to actually quantify how much of the deletion you have, the heteroplasmy level of the mitoDNA deletion, because it's not reliable just from the next gen sequence alone, because it preferentially amplifies smaller deleted molecules and it could be misleading. We also look at histology, immunohistochemistry, electron microscopy, and a lot more. Hopefully you'll hear about in future lectures. It is a lot to know. None of us can keep all of these genes and potential spectrum in their heads. And so working with colleagues from around the world, we created a mitochondrial disease genes compendium, which has both an electronic version and a book. So you can be in clinic or in the lab and look up a given gene and understand the spectrum of clinical problems, the therapeutics that might be available, specific support groups, or research for currently there's over 250 genes and more are being added. And what's nice is that you can also link to a website I'll show you where we have information on each variant. So that's up to date and you can be hooked in to know what's the likelihood any gene is associated with a specific syndrome or a variant. And this replaces our older way of thinking about it, that a patient to have a mito disease had to have one of these acronyms, MELAS, MRF, which is epilepsy and ragged red fibers, NARP, neurogenic ataxia and retinitis pigmentosa. We now realize that almost all of those syndromes have multiple causes. And so a more useful way for families who are trying to understand, what to expect for their child or themselves, or what the recurrence might be, or the syndromes to look out for, or the symptoms to screen for, is to think about what the causal gene is. This also reminds me to take you to a website that's free that the United Mitochondrial Disease Foundation has sponsored for the last 9 years, 7 years since the website went live, and it's used around the world to both help clinicians or bioinformaticians who are geneticists understand the data that you might have on a patient, or so a given variant or a whole exome data set or a mitogenome to analyze it yourself, understand the haplogroup and so on, or browse the information to understand what are the features of a certain syndrome. So we define things by a common ontology, a common data dictionary, the human phenotype ontology, and you can then say, what are the terms that make up MELAS? What are the terms that make up Lee syndrome? What are the genes that cause it? What are the variants? That's some of the most popular uses. And you can see that we currently include information on more than 183 clinical disease definitions and more than 1,500 genes. I actually think it's over 2,000 now. There's also lots of information about browsers and other activities. So I'd encourage you all to log in and utilize it, and reach out if you have any questions. This is an example of if you have an exome data set, which now is very possible to get from the research or clinical lab, you can put in the doctor's note summary or the terms and annotate them to HPO terms, sensory neural hearing loss, conductive hearing loss, add the file, and within minutes, they'll download to you a file with every inheritance mode and what was the likely genetic and variant cause of that symptoms that you put in that patient. And about 70% of the time, the top hit is the answer. So this could be a useful resource for you in clinic if you have access to the data. I'd like to switch in the last few minutes to therapies. In the past, people would say, why do we have to diagnose this? We can't help these people. I think it's very different now, and we recognize there's a lot of ways to potentially help patients with mito disease. I love this review. If you want to read one to start, the Nature Review Disease Primers by Dr. Grenier-Gorman et al. From 2016 is outstanding. And this is a really helpful way to think of the mitochondria now as a summary of what I've shared with you, that there is the electron transport chain in the inner mitochondrial membrane, and there's not one copy of it. It's really many, and they're not really separated. They're all on top of each other to improve electron flow. But when it's not just the electron transport chain, it's bringing nucleic acids in. It's taking the mitochondrial DNA, maintaining it, replicating it, right, to make more, translating it, and transcribing to make these polypeptides. It's also, once you make ATP, translocating it in and out of the cell. There's out of the mitochondria to the rest of the cell. There's phosphate carriers. There's actually more than 100 transporters, because the inner membrane is very impermeable. So there's more than 100 transporters to bring different metabolites in and out of the mitochondria and different metabolic products. So there's just a lot going on. And I'll give one example. There was a famous case a few years back of a little boy named Charlie Gard that was all over the media. The Pope and the president were tweeting. It was a child in the UK that wanted to come to the US for therapy. And everybody had an opinion, and there was like a GoFundMe campaign. They raised millions of dollars, and the supreme regulatory body in England said, no, the child cannot go. So a lot of people had a lot of opinions. But if you look back at what that child had, he had a defect in bringing nucleic acids into the mitochondria. And the therapy that he was being offered was simply, if he were to come here, was simply a form of the nucleic acid that could bypass the defect. And so that's a very different therapy than for somebody who has an inherent problem in their electron transport chain. That child's electron transport chain was fine, but they couldn't maintain their mitochondrial DNA pools. They had something called mitochondrial depletion. They didn't have enough of it. Over time, there's been a mitochondrial medicine society forum. I'd highly encourage you to look at their website. It's also, I think, open to people with a ton of information. And a lot of the people in this SIMD have participated to really create expert guidelines, like about exercise. How do you manage acute stroke? What about anesthesia? Some mito patients are hypersensitive to anesthesia and won't wake up after on time, or will have a really prolonged emergence from anesthesia or neurologic problems after. How should we handle this? What about acute illness? What about vitamins and supplements? And so there's a lot of consensus information and a lot of guidelines now available to you to standardize the care for patients. There are no FDA-approved therapies or cures, and this is because these are individually rare disorders, like we just went over. More than 350 genes in 2 genomes, more than 16 symptoms per patient, no universal biomarker, and the therapeutic options have been limited. It doesn't mean there's none. It just means that they're limited and they haven't been well studied in clinical trials. But some of them have, and the one therapy you'll get every mito doctor to agree to is exercise. Both aerobic exercise and isotonic exercise work that's come a lot out of Dr. Mark Tarnopolsky's group, showing that you actually can make more mitochondria. And remarkably, if you do weightlifting, when you tear and repair the muscle, you actually can shift the heteroplasmy of a mutation back towards the wild type. So really remarkable effects of exercise. And a lot of our patients are also very deconditioned. So it actually exacerbates their problem. And so to the extent that it's safe and tolerated, from a cardiac and renal perspective, exercise is very encouraged. And if you look at a family where there's multiple affected individuals, those who exercise are often much more healthy and do better. Nutritional therapy is also really important. I told you from the beginning that the mitochondria are sort of the intermediary metabolic, conductor of the orchestra. So if they're not using aerobic respiration properly, their only other way to make energy is through glycolysis, and it's not as efficient, but it is more upregulated in them. We'll talk about this more in a few minutes. There's probably a lot more we need to learn about different types of mito disease and different nutritional strategies. There's been a one-size-fits-all cocktail, often called a mitochondrial cocktail, with a little bit of negative implications to it. People say, well, a cocktail is good for a party, but it's not good for a clinic, right? Well, over time, a lot of smart people would say, well, this enzyme is deficient. This is a good cofactor, or this metabolite, could help improve the process. And so there have been many case reports of people that have been improved. Some of it has been hard to test in a clinical trial. About a decade ago, several of our colleagues tried to create a coenzyme Q trial, and it wasn't successful because patients would not enroll because they did not understand why they should enter a trial for CoQ where they may not get it when they can just go to the pharmacy and pick it up themselves. So some of this is that once these are widely available, it's very hard to form a trial, but it doesn't mean they do or don't work. It means we don't know. This is what we published last year. Dr. Barcelos was one of the NAMDAC fellows, along with Dr. Shadyac. We now have a mitochondrial medicine fellowship program. If you know anybody who's interested, it's a one-year training program. And we're always looking for great people who are interested to participate. And they did a wonderful review. We hadn't reviewed this since 2009. What types of medicines do we give? And so if you use this Current Opinions in Pediatrics article, you'll see that most patients, we recommend a multivitamin and a B50, not a B100, because that's so big, people can't swallow it if you've ever looked at it. Most people are on CoQ, even though primary CoQ deficiency is very rare. And most people are on some form of an antioxidant that's pretty safe and well tolerated. So typically vitamin E or alpha lipoic and biotin. And we think of these primarily, we tell families like paper towels, if there's too many free radicals. And Dr. Greg Enns and others have shown there's way too many free radicals in most of these diseases, that these help you scavenge them. Vitamin E mainly in the cytosol, lipoic acid mainly in the mitochondria. And then we give biotin because lipoic acid has been reported to result in low biotin levels in some cases because it requires it as a cofactor. And then depending on the type of other problems going on, we might add other therapies. For example, we often now measure glutathione levels. That's your major antioxidant scavenging enzyme that recycles itself. It has a reduced and active form. And in many patients, glutathione levels are low. And there's now multiple labs where you can get this tested. When glutathione is low, we give N-acetylcysteine. Why? Because glutathione is made up of 3 amino acids, cysteine, glycine, and glutamate. And so giving N-acetylcysteine helps the body generate more glutathione. We tell families it's like in Pac-Man, when you run out of Pac-Man and you need more lives, and NAC can help do that. And we do see the levels rising. But again, we try to get them into the normal range. So we're trying to be more objective over time and give therapies based upon specific symptoms. There's also been reports, this is by Dr. Holger Prokish's group from 2017, showing that if you have a specific genetic cause, there might be reason to give specific therapies, potentially at even higher levels in some cases, with different treatment responses than we might use in the standard medicine approaches. So these are really just to say that once you know what's going on, you can have a lot more, I think, intelligent design of precision therapies. I'll go very quickly through this so we can get to questions, but we really don't know the right way to feed people with mitochondrial disease. We know that a lot of them don't eat enough. We now do resting energy expenditures in people, and we realize that their nutritional intake is often too low for their energy expenditures. Some people are overweight, some people are underweight, but optimizing nutrition can really go an enormous way to make people feeling and functioning better. In little kids, like with those with Lee syndrome, or even older ones with Lee syndrome, we very quickly move to supplemental feeds if we need to. So gastrostomy or total parenteral nutrition if their gut doesn't work. Because if you have a patient undergo catabolic stress and fast, when they have a mitochondrial disease, they're at high risk for neurodevelopmental regression and progression of their disease. So optimizing nutrition, preventing fasting can go a long way to helping making people feel better. And these people are at high risk for problems like reflux and dysmotility, for stroke that might prevent them from swallowing properly, and so on. We also recommend looking, and we have a dietitian evaluate every one of our patients for micronutrient insufficiencies. And again, we avoid fasting, encourage frequent small meals, recommend never fasting, and really encourage fluid intake, especially in heat and under conditions of stress. Amino acid therapies are definitely worth knowing during your residencies and fellowships. Arginine and citrulline have long been known for urea cycle disorders, and Dr. Koga in Japan around 2005 made a really seminal report that they were useful for acute stroke treatment in MELAS. Over time, there's been debate whether in adult MELAS that is effective in the short term, meaning in the 2 hours post infusion. But over time, he's reported long-term data, and it does appear to improve long-term survival. I think he reported 9-year data. We also have looked back at our own studies where we've looked at the acute or subacute use of arginine in other diseases besides MELAS, and we had about a 50% clinical recovery in people who had had hemiplegic strokes by discharge for mitochondrial metabolic stroke. So there's very little risk to using arginine. The thought is that it's acting as a nitric oxide donor to target endothelial ischemia within the microvascular vessels, and that appears to be part of the etiology of metabolic stroke. So we typically use a standard dose prophylactically to try to prevent stroke, typically starting with arginine, moving to citrulline if it's refractory. Dr. Scalia in Texas is actually doing a study comparing the two in acute stroke. And in acute stroke, we typically give arginine, again, for IV, where it's much more potent, and we give it for up to 5 days to try to reduce the sequelae. In terms of the macronutrient profile, there is no clear recommendations of protein, carbs, and fat to use. Some people recommend using the ketogenic diet, and in subtypes of mito disease like pyruvate dehydrogenase deficiency, it's the gold standard. But that said, ketogenic diet in people with respiratory chain problems is very controversial. It's known to increase ketones and succinate, the starvation response. There have been some, in mouse studies, there are some mice where it slows disease progression and other mice where it exacerbates their progression. So it's not consistent. And in patients who often have high triglycerides, who often have impairment of fatty acid oxidation and PPAR, the diet's often not tolerated, and it can be associated with long-term health risks. There was a really important study a few years back by Dr. Sumalanian's group in Finland, where they had seen a good response of the Atkins diet in people with depletion or deletions in the mitochondrial DNA due to things like twinkle deficiency, which is the helicase that helps unwind the mitochondrial DNA. They did a small trial in patients. They picked 5 adults with deletions and 10 healthy controls, and all they did was change their macronutrient ratio. So they went from a high carb diet to a very low carb diet. And when they did this, the healthy controls had no problems. They handled it fine. But all 5 deletion patients had to stop the diet within days to a week or so due to severe muscle pain, burning that progressed in the same fashion in all of them, from their legs to their back, to their arms, to their neck. They got terrible headaches, increased tired, and they did muscle biopsies that showed there was necrosis of their muscle fibers, their CK went up, and so on. So it's to say that nutrition has a big effect on our metabolism, but in mitochondrial disease, it may not be handled the same, and that clearly it's having physiologic changes that may not be tolerated. That said, to get people ketotic, you have to give high fat. And that seems to be the problem in mito diseases. That's not tolerated. But there have been other studies over time that look at the components of the ketogenic diet that suggest maybe those, if you can give them directly without giving high fat, would be tolerated. And so there's been studies on triacylglycerol, triheptanoin, decanoic acid, and so on. And so these have potential for the future. Another thing to think about is, our patients crave carbohydrates. Most people I know can go to noon without eating breakfast. A mito disease patient, if you ask them, has to eat within minutes of waking up. And sometimes they'll tell you the craziest things they eat. They have to eat a soft pretzel. They have to eat carbs or potato chips. Why? Well, they're craving carbs. They feel like they can't think clearly. They have no energy until they eat. There's been a lot of controversy, just as uncertain. Should we be giving glucose? What's the glucose infusion rate? Can they handle it? There are some types that can't. There are many that can. A lot of the therapy we do for patients when they're having an intercurrent infection or some sort of stress is to give them fluids with carbs and just monitor how they handle it. But a lot of mito patients feel a lot better when they're on carbs. And so just to get towards the end, the respiratory chain is complex. But when we think about it more simply, the respiratory chain makes things. It makes some products: ATP, free radicals, nucleic acids, NAD. Complex one is NADH dehydrogenase. It converts NADH to NAD. When these things are disrupted, right? Too many free radicals, too little ATP, et cetera, we have other therapies. And so we've been modeling these in animal models for quite some time. And often a combination therapy is more productive and synergistic to counteract the deficiencies that occur when the respiratory chain doesn't work properly than any one alone. A lot of the therapies think about the mitochondrial problems that happen and trying to get therapies into the mitochondria when there's genetic changes or environmental changes. But our group and others have realized when the mitochondria doesn't work for whatever reason, a drug, a toxin, a genetic problem, the cell knows about it. And depending on which organ and how much demand it has, under what stress and how much supply it's getting, it's responding. It's responding with transcriptional changes, translational changes, and certain signaling molecules are becoming activated. We call them the nutrient sensing signaling network. You'll hear about them in many other diseases, mTORC, AMP kinase, sirtuins, PPAR, FOXO. They're trying to rebalance the energy supply and demand. So what we've realized over time, and there's a lot of clinical trials now emerging in these domains, that you can actually modify these signaling pathways themselves, and that might have therapeutic benefit in mitochondrial disease. You haven't changed the mitochondrial problem. You haven't changed the genetic disorder. What you've done is change the cell's response to it, because the cell's response might be contributing, even though it had good intentions, presumably, it might be contributing to the clinical disorder and dysfunction. And so there's trials coming out with mTORC inhibitors, PPAR modulators, sirtuin agonists, nutritional therapies, antioxidants, and so on. So it's a really exciting time in the field. And in the future, we need to figure out which patients get which therapy under which conditions, and do we use their own cells, for example? Dr. Van Hove, who you know well on the call from Colorado, thinks a lot about how we can better understand mito disease in patient cells, and can we also use animal models? The goal would be is that when we're designing clinical trials and defining diseases and prioritizing outcomes and working on treatments, that we wouldn't just jump to clinical trials. The problem is this happens a lot, and 95% of the time, clinical trials fail for all diseases. So what we and others propose is that we should be looking at the proposed therapy in a model, whether it's the patient's cells, whether it's an animal model. We should make sure that it's tolerated, that it doesn't kill the model or the cells, and that we understand which types of disease might respond, and then design better, more cost-effective precision trials where we get responders and leave out people likely to be harmed by the therapy. In our world, we use C. elegans, zebrafish, mice, and human cells. We test therapies that people have purported should work, empiric therapies that we just try and they seem to work, drug libraries, genetic libraries, and we try to see what improves survival, function, and feeling. That's what the FDA will need to make a therapy go forward. They don't care if a therapy changes lactate. They want to know that a therapy improves somebody's life. And so that's really the future of mitochondrial disease, is understanding the cause so you can actually get to a therapy. At our center, we work on optimizing patients' care, using the types of diagnostics we discussed, and optimizing their health, as we talked about per the guidelines, doing clinical research to better understand the patient's cells, and to understand what outcome measures we should be checking on, and then working in the translational setting, to better understand novel genetic causes of disease, validating those, developing therapies, partnering with companies who can actually bring therapies to these patients, and so on. So to conclude, I hope you agree, mitochondrial disease is highly heterogeneous, but it is recognizable. And especially with better testing, we recognize there's extensive variability. You just have to get comfortable with the fact there's a lot of variability, allelic and locus genetic variability, phenotypic variability, and you have to be vigilant. You have to look for the problem so you can help. In terms of the testing, molecular diagnostic testing in 2021 is essential. When you suspect mitochondrial disease, you can't look at somebody and know how their mitochondria are working. You need to do the testing, and you need to think about both genomes. And finally, it's really important to become familiar with the standard of care guidelines so that we all begin, to take better care of our patients. And I think with that, I hope you're excited that precision mitochondrial medicine is possible. So there will be some one-size-fits-all approaches, and there will be a lot of individual optimization based upon who they are and what the cause of their disease might be. I will gladly take any questions. Thank you.
Thank you, Dr. Falk. That was fantastic. A really, really good and thorough review of mito disease. And love the articles you referenced. Those are some good go-tos, I think. There were some questions that came up in the chat that I think we could probably address straight off. Dr. Calhoun asked, and Amy, I don't know if you wanted to speak up. I can read the question though. Basically, she was wondering, how do you distinguish symptoms in adults, especially when symptoms are things like fatigue and muscle weakness and things like that, when those are common problems in older adults anyway, from true primary mitochondrial disease? And I would kind of piggyback onto that. Has your threshold for initiating a workup changed at all with wider availability of molecular testing?
Yeah, that's a great question. So typically fatigue is not the isolated problem, right? So we don't really, this is Dr. Goldstein. She'll be the first to say, my partner, we don't really primarily see people with chronic fatigue. Typically it's fatigue with other problems. In the past, we would say you can never end the search for mitochondrial disease, but pretty much now you can do genetic testing, biochemical screening, and if you really have suspicion, a muscle biopsy, especially in adults, the muscle biopsies often show histologic or electron microscopy abnormalities. And so we're much more likely to use a muscle biopsy in an adult than we are in a child. And there's a couple sites, ours and UCSD and Dr. Tarnopolsky, where you can get a needle muscle biopsy where you don't need to have anesthesia. And so you could do it in the clinical setting. And that's really nice. Sometimes we get people where they're on like statins, and that's probably a whole other lecture. Statin myopathy occurs in about 3% of patients. But statins prevent coenzyme Q production, and they cause mitochondrial depletion to about 30% of control. So 2-3 times a year, we get an adult with fatigue and myopathy, and we realize they're on statins. We ask that they come off. And they might be on statins because of a family history of heart disease, no actual problems in themselves. We ask that they come off, we'll repeat the biopsy, and we'll see that the mitochondrial DNA amount goes from 30% back up to 100%. So you definitely have to think in older people on polypharmacy. Some of these medicines are really inhibiting mitochondrial function. The other thing that we use is we've been developing a lot of non-invasive tests to complement the muscle biopsy. So yeah, certainly doing mitochondrial DNA testing in an adult makes sense. 2/3 of their diseases are mito disease. Mito are mtDNA disease. And we can do the testing out very thorough. So we might start non-invasive, move to muscle biopsy a little quicker, especially if they're having another procedure, or do it in the clinical setting as a needle biopsy. And just as an example, Dr. Tarnopolsky's published reports of doing more than 3,000. So they're really very readily doable, I think, if you get clinically credentialed to do them. And then we would do CPET. We would ask them to go to the exercise physiology lab and have cardiopulmonary exercise testing. We're developing some pretty cool new tests. We have an MRI-based test now where they can do phosphocreatine imaging. They can look at not just their creatine levels, but how their phosphocreatine, the high energy bond that's in muscle and other tissues, we can actually quantify how quickly they use it up after exercise, and how quickly they recover. And the rate of recovery, 100% depends on oxphos. That's an MRI test. And that's something that we'll now do for people that just became available. We're also developing other sensors. We realize that are non-invasive. We realize these are big questions, and that once we have these better tests, we'll probably start to figure out, well, maybe chronic fatigue is a mito disease. We just need better tests to get there.
Thanks. The next question is from Kylie James, one of our LGG fellows, and she's asking about embryonic heteroplasmy and if there's selective pressures in any particular progenitor cells or organs to select for or against higher or lower heteroplasmy.
Yeah, that's a really good question. So people like Patrick Chinnery and Hubert Smeets, they have some really great papers on this topic that I would definitely refer you to. It's really interesting because like the NARP mutation, the ATP6 mutation actually escapes. There's some sort of early, the QC protocol that happens in early embryos. So at some point, the oxidative phosphorylation system needs to be working in the embryo. And because it's in complex 5, it actually changes, I think it's the membrane potential. The membrane potential is like falsely high because the problem is that it's not being dissipated and it gets through this quality control check in the early embryo. So it's fascinating why you'll see people with NARP have homoplasmic mutations. Like, how did this get through? So in the last year or two, there's been some really cool kind of discoveries about that. Patrick Chinnery is probably the leader, though, if you start to read his work and sort of really dissecting the nuances of that very complicated question. Probably more than I can do justice to now, but I'm happy to talk about it later. I don't think we know everything, but clearly there's some checks and balances that happen around day 3 to day 5.
Dr. Mori had a really practical question. Do you know in the US where we can send urine for testing?
Yeah, so we actually pressured GeneDx to do it. So the problem isn't that urine doesn't have DNA, it's that the quality of the urine sediment is really bad. And so the DNA is often fragmented and disrupted. So I think Baylor will do a targeted mutation validation. I'm not sure, not Baylor, they used to, but now they don't. Dr. Jing Wang, who was doing this at Baylor, moved to our institution. And so CHOP will now handle urine. In terms of, will they do the full sequencing or validation of the mutation? She'll definitely do the validation of the mutation. I think they use long-range PCR products. Again, if you talk with Jing Wang, I can put her name in the chat. She's terrific, and she'll tell you exactly what they will and won't do. On our website also, you can find more information about it. But this to us was a really big deal. We couldn't get urine testing either, so we had to set it up.
It's really good to know. And again, super practical.
And the other thing I'll tell you about Jing, what she did, we always had a really hard time getting insurance coverage for the mother. It's really important if you find a variant to know if it's inherited, but it's very hard to get an insurance company to understand that. And so then you go years and you're like, I have a variant of uncertain significance. I wish I knew what it was in the mother, because if it was higher in the mother and the mother's healthy, you might think it's meaningless. Whereas vice versa, if it wasn't there, you might be more excited. So the test, yeah, I think GeneDx does do targeted testing. CHOP will definitely do targeted testing. I'm not sure if she'll accept it for untargeted. I have to ask her. But what she did do for us is that if you send the mother's sample at the same time as the kid, it's included for full next gen analysis. And that's pretty cool because otherwise there's like no way to really get it done. And you don't have to send the same tissue. You can send like muscle on the kid and urine on the mom, or blood, she doesn't care. So to me, that's like it's worth knowing Jing because she'll help you out. But again, it's included with the test. So you don't have to do it. You don't have to send it subsequently. And she'll also give you I think we have the only rapid report. You'll get a 2-week answer if you ever really needed it. And I think a verbal in 1. So we agree, this is an important chromosome and we need real methods to help us analyze it reliably.
Anybody else have questions for Dr. Falk before we go? It's 1:29, so we're pushing the envelope on time. Well, thank you very much. I encourage everybody to fill out the survey at the end of each session. Thank you very much, Marnie, for coming to talk with us. This was, again, a great overview and a great review of the most current literature. So thank you very much. And we will see you guys again next week, where we will have Dr. Nick Amu talk about amino acid disorders, specifically urea cycle. So thank you, and we'll see you all again next week. Bye.
Terrific. And remember, if you want to do a fellowship, reach out to me.
Yes.
if the mito bug has bit you, we're happy to consider you. All right. Take care, everybody.
Thanks, Marnie. Bye-bye.